I'd like to invite all our speakers to the stage. We're going to take questions from the audience. There's a roaming mic, so if you'd like to ask a question, raise your hand and the mic will come to you. Okay, I see our first question already.
Hi, Peter Galadin from Stanford Cancer Center, South Bay. The Shiva trial published late last year was a proof of concept examining, I suppose, a histology agnostic approach to treatment using molecular data. Not sure how much of that was technically genomic-based data versus other types of molecular testing, but it did not show a progression-free survival advantage. These were advanced cancer patients, multiple prior lines of therapy. I don't know if you know about the trial or if there were lessons that could be learned that maybe are shortcomings not seen in the I-SPY or 2 or in the upcoming or proceeding right now NCI MATCH trial.
Should I answer this? I'm not completely familiar with the trial you're referring to, but I can maybe say that for the I-SPY trial, I think one of the advantages is that we pre-selected patients, and these are early. So I think one thing is that these are early diagnosed patients, whereas the NCI MATCH trial is mainly in patients who have metastatic disease. So I think the advantage to look into the early diagnosed patients is that you have, first of all, a chance to actually cure patients, or at least make them much longer tumor-free. And tumors are not as derailed, potentially, as in the metastatic setting. Secondly, I think our pre-selection for patients that are at very high risk for recurrence also allows us to really monitor for an effect size that is meaningful. And the thresholds to graduate a drug is actually really, really high. So the difference needs to be at least 20-25% on average for a drug to graduate. I think that's maybe. I think it makes sense to really look into the biology of the disease. As was said before, there's heterogeneity, like between patients, but even within a tumor, but also between patients and their tumor type, to understand that your denominator becomes as clean as you can get it.
Any other questions? Okay, I'd like to follow up on this issue of tumor heterogeneity. There's no doubt that there's so many advances in the technology and the deep profiling and bringing awareness to patients and enthusiasm support by patients. And yet, this is a very complex problem, and we still have a lot of progress to make. And so this heterogeneity question, we're looking more at it now in the context of immunotherapy, which is very exciting. But there's many other aspects of heterogeneity, and I'm just wondering, in the context of your own work, how you think of heterogeneity, and do you really think it's important to address it so that we can make progress? Laura, you want to start?
I can start. So I do think heterogeneity exists. I think in some instances, you're able to shrink the tumor by maybe a combination of therapies, but you're never sure that there might be one subclone that might be able to stay, and that may actually exceed and will be the one that will repopulate elsewhere in the body. So I think that the trials, how they're done right now, are by looking at a group level. I think we much more need to go and look into one individual patient to really understand what is the response that we see in a particular patient. So do we see remaining tumor cells? And I think the whole new capability to do cell-free DNA or cell-free RNA testing, I think will be of use to understand what is the residual tumor burden after an initial treatment. So I think it exists. I think we can derive knowledge from looking into a group level, and with our approach you can zoom in, as I said, to get the denominator as clean as possible. But even then, I think there are differences, so you need to monitor within a patient.
I would like to add to that. A genetic interactions-based approach is actually very attractive for targeting heterogeneous tumors, because you find some genes that are synthetic lethal hubs. That means that they have many synthetic lethal partners in specific tumors. Okay? Now, if you can target these genes, then even if the tumor is heterogeneous and different subpopulations of the tumors have inactive partners, different inactive partners of the same hub, by targeting the hub, you can hopefully kill a lot of the subpopulations in the different heterogeneous subpopulations in the tumor. I hope you get me. Synthetic lethality-based approach has also another very attractive potential feature once we will learn how to harness it. And that is, if you think about it, the more the tumor becomes more aggressive, the more there are genomic abnormalities. More genes are lost or increased in the copy number, or the expression is shut down, or they are mutated. The more the genomic landscape becomes more abnormal, the more of the potential synthetic lethal partners, or actually synthetic dosage lethal partners, if you're familiar with the term, will become inactive or overactive and lend themselves. The tumor will lend itself to such genetic interactions-based therapies. The more aggressive it is, the more vulnerable it comes, and this sense and exposing new vulnerabilities and opportunities to target it. So heterogeneity and aggressiveness is good for us in a paradoxical manner, of course.
Richard?
Yeah, so I agree with what's been said. I guess the only thing I would add is specifically for things like neoantigens in immunotherapy, it's clear that the heterogeneity is actually very important in terms of figuring out which neoantigens are likely to be predictive, for instance, of response. So, for example, there are recent studies that have suggested that the truncal mutations, or the mutations that are higher frequency in tumors that are predicted to be neoantigens, those are more important for ultimately predicting response than, let's say, the ones that are subclones at low frequency. And so that's a good example of where, depending on what you're looking at, in this case, if you're looking at immunotherapy, understanding the heterogeneity can really impact the decisions at the end.
Yeah, I think study of heterogeneity is very important. Tumor, and it's basically a consequence of how tumor develops. Tumor should be thought of as a unicellular parasitic organism evolving within the host's body. And if it is seen as such, its heterogeneity and its function should be to a large extent predictable, because let's remember that within the development of a sizable tumor, we have tens to hundreds of billions of cell divisions. So the entire mutational landscape of the human genome is essentially sampled. So I think as we study tumors more and their heterogeneity, we will see that things are more and more predictable. Complicated, yes, because each tissue also has a different environment which influences this evolution and function, but ultimately predictable. And that's why I think Eitan's direction of finding how to stop this evolution, this parasite in its tracks, with things like synthetic lethality, and thinking in a rational way of how to eliminate all the different subpopulations will be more and more important and promising.
Great, thank you. So I want to turn to the audience, see if there's any questions from the audience. Yes?
Hi, Daniel Markowitz, Swedish Cancer Institute. Just had a question. Have any of many of you are working on very sophisticated testing. Have you done any of this testing longitudinally over time in single patients? And to be more specific, if you have a patient who's developed a resistant tumor, are the cells that have developed farther distance away from the original tumor, or greater distance? Are the things that are taking the patient down very similar but simply mutated in a small way, or are these part of the heterogeneity that's grown up like an ill wind from the distance and has taken the patient while we've been busily treating in another direction? And this question actually has to do with an interaction between some of all of your testing.
Yeah. So, thank you for the question. Two comments on that. One of them, I haven't shown the data, but we analyzed based on these inferred molecular resistance pathways that we delineate. We analyzed the state of progression of resistance in breast cancer patients as they advance in the disease. Okay, using the Curtis data and stuff like that. And the picture is rather frightening. You see a continuous monotonic increase in the resistance pathways across, 2,000 breast cancer patients in all subtypes and so on. The tumors become more and more resistant, and you actually see this molecular fingerprinting of this resistance. In another study, we analyzed, there's a Nature paper last year where they sequenced patients after the resistance has emerged in the tumors. Okay, so they were treated, some responded, and among those responded, some have emerged resistance and others have not. And they sequenced them and they looked on the differential signatures. And we analyzed that, and we can see that those that have evolved resistance, they either evolved resistance by activating the pumps and throwing the drugs out, or they evolved resistance by activating these molecular resistant pathways. So you can see the wide array that cancer cells have to develop resistance and their smartness. You can see a marked, strict inverse relationship, and they are very economical. They don't do both usually. If they succeed to throw the drug, then they are fine, and if not, they go on and change the regulatory systems and evolve these molecular resistance pathways and so on.
But I think maybe the interesting underlying part is, or maybe that those things occur, but there are also, I think, with the breast cancer intrinsic subtypes as developed here at Stanford for breast cancer from luminal to basal type. I actually did a study with Chuck Peru once to see if you had patients with primary disease, metastatic disease, how do these prognostic signatures or subtype signatures that we had developed, how do they hold up? And those are actually very much the same. So there is some sort of a hardwiring that continues to be the same, and on top of that...
Absolutely. Also in our analysis, the hardwiring pathways, I wasn't clear, sorry. The hardwiring pathways are the same, but we see that they become more and more activated as the tumor progresses. So it's, in the line.
So the treatment probably needs to hit both to be effective.
Totally.
Okay. There's another question? Yeah? Ah, terrific.
Hi, this is Ram Akela from University of California. All of you have started alluding to something I've been thinking about for a while, and maybe we can start with Seraphim and continue. The question seems to me that you're thinking of a dynamic evolution, right? So the question is, think of more dynamic models rather than static models if you think of a progression, right? So when you use the machine learning techniques, they're typically more static prediction techniques, but if you think of Kalman filtering and so on, you're more dynamic techniques. So I'm sort of curious, if you think of, but cancer and typically cancer immunology and so on, you have more of variation. But as you think of clinical data, you have much more variation. So I'm sort of curious, given the sort of problems you're describing, adaptive methods or progression of disease and so on, right? How much of a sequential learning or temporal model might be more effective rather than static models is a question I have for all of you.
These are good points. I think that the progression of cancer, in principle, at least the way I like to think about it, should be thought of in terms of some stochastic but somewhat predictable model. So basically from a state A of a given collection of cells, what is likely to happen if something is applied to them, such as a drug treatment, maybe they will get to state B. So we need to understand those things. So as Eitan has shown in his talk as well, like drug resistance can be predictable as well. So in general, any type of evolution within a set of cells that are replicating very fast should be, that's our goal, to basically be able to predict for a given collection of cells that we know are growing in a tumorous way. We want to be able to predict how they will evolve, what are the likely pathways. So we know that if we apply this drug, then maybe it will kill them, but some of them will evolve to be resistant. If we apply the other one, maybe that will happen if we apply both, or if we do this other thing. So we want to be able to understand those statistical facts, basically. So we are not there yet, but that's what we are trying to do.
I want to continue on what Seraphim said, and said that your ideas are very nice, but there's some problems. So if you want to analyze it with the clinical data, so yes, this is a conference for big data, and there is big data, and the data is increasing, but it's just big. It's not very large. So, for example, I come up every morning, I have another idea, but mostly I trash them because the data isn't there. Okay, so this is a wonderful idea, but simply the data that you would like on the clinical level to study the question you have raised isn't there. Okay, the data is big, but it's not very large. So hopefully in one of the next conferences, it will be called the Very Large Conference, right? Hopefully. And it is exponentially growing, so there is hope, right? And this will change things. Currently, for many of the questions that need to be studied, let me make it clear, the data isn't there. Now, you can study these questions in cell lines, okay, and so on. But let me tell you, this is something that we are studying in depth in my lab. The relation between what you find in omics analysis in cancer cell lines and so on, and even in animal models, and what you find in the clinic is far from desired, and this is the understatement of the year. Okay? And this is a very, very serious problem, right? There's a whole branch of research analyzing cell line data molecules with claims about the clinical significance, and if you look at it very carefully and rigorously, it's quite questionable.
Maybe there are exceptions.
Yes, of course.
I'm an optimist.
Example, studies from my lab are total exceptions to that.
There's no doubt that the cell line and mouse models are incredibly relevant to understand underlying biology, because right now, to get at mechanism, we really need experimental systems that we can functionally interrogate. And so, but do they capture the full complexity of human disease? No. Are they giving us insights? I think the answer is yes.
They're the
But
best.
maybe I actually wanted to give the example. So my experience in this breast cancer treatment, that it seems that the treatments that are targeting the DNA repair are actually more uniformly effective than the ones that are geared towards a AKT/MEK pathway, because there are so many side branches and resistance pathways that can circumvent the target. And it seems that for DNA repair inhibition, it's a bit more, you hit sort of the hardwiring, maybe, so to
It's a very
speak.
conserved, also, it's a very conserved mechanism and pathway, so it's more likely, as you say, that the
So to
studying-
understand which ones are those, I think will help us.
I think there's a new question.
Zina Good. I'm a PhD student in immunology here. And my question is about selecting the right drug for the right patient. We know that in cancer immunotherapy treatment are successful if you are able to reduce the tumor burden. So we need really effective drugs. But I was wondering whether there are existing efforts, or if not, what kind of data are we missing to rate the drugs based on how immunostimulatory versus inhibiting the immune systems they are. Because you probably want to remove the tumor burden while not inhibiting the ongoing immune response.
think
Yeah. So,
go
so
for you.
I think I think I mentioned this a little bit in my talk, but I think this is early days in understanding how people are responding and what are the immune modifiers, especially for people that are receiving immunotherapy. And it's complicated, and because if you talk about systems biology, the immune system is probably the best example of systems biology in action with all these different players, with the T cells and innate immune system and such. So I think it comes down to we need more studies that actually get the right data and track the right data with outcomes and also being tracked. So that patients that had this kind of signature, expression signature for these kind of immune pathways tended to do better or worse on treatment. And those studies don't really exist right now. But I think that the getting the comprehensive characterization of sort of the tumor, the immune response in the tumor, and collecting that data in these trials that eventually lead to outcomes, I think, and putting the two together are going to give us the insights to make better decisions about therapies ultimately. And those are the types of things and studies that we're trying to get involved in.
But maybe a good follow-up question would be, like in a number of tumor types, like breast cancer, in early diagnosis, you have the opportunity to do so-called neoadjuvant treatment. So you take a biopsy of the tumor and then leave the tumor there so you can monitor a response. And so that gives you, I think, a good measure of, for that particular patient, whether the treatment works. So I think that also took, and there in some tumor types that also exist. So I think it also is an effort, I think what you're alluding to, that we need to exactly know what the endpoint is and how to define the tumor burden. And I think their imaging is probably one of the areas to further explore, to really understand what are the differences in tumor size that would allow you to foretell something about the longer term outcome.
Yeah, and I think in immunotherapy it's tricky because we know that tumors can increase in size, temporarily before they decrease in size. And so defining those endpoints and tracking that has been one of the challenges. And in fact, in the earliest studies, I think there was some disappointment with the results because they used traditional methods to kind of measure, response to these immunotherapies, and they thought they were headed in the wrong direction. The tumors were increasing in size, and so some trials, had potentially, it looked like they had bad outcomes, but then they realized that, they were tracking the wrong endpoint. So I think your point is exactly correct. I think the measuring directly the tumor, the infiltrates, the immune infiltrates in the tumor is maybe one of those biomarkers that could be very helpful for that sort of thing.
Okay. So I think with that, we will wrap up this session. Please join me in thanking our speakers.